Good afternoon, everyone. Welcome at the second day of our dispersal-related trade colloquium. My name is Renske Onstein, and I'm going to moderate tonight. We have 4 talks coming up. So yesterday we spoke a lot about, what is dispersal, the basics of the dispersal process, what kind of work has been done in dispersal from both ecological, evolutionary sides, and we went through a lot of different systems, terrestrial systems, aquatic systems, different plant groups, etc. And today we're going to continue with that line of thought, starting first with understanding the importance of using trade data when we look at dispersal. Then we're going to hear about plants and the biotic and abiotic strategies they have evolved for dispersal. Moving on to fungi, very excited about that. I think it's a lot less known how dispersal works in this group. And then we're going to again take a more evolutionary focus, move to amphibians and hybrid zones in dispersal. This colloquium is part of a workshop we're having this week with several people, among them the speakers, but also people more remotely. So I hope they're all online, as well as other people online, so welcome as well. You will be able to ask questions in the chat on YouTube. I will repeat that later on, and hopefully we can keep track of that and answer your questions as well. And of course I would like to thank the funding for this colloquium. That's by the Journal of Biogeography, as well as Naturalis, that allowed us to be here and talk about dispersal together. So with that, I would like to introduce our first speaker, Adriana, and she's going to talk about the importance of using the right dispersal trade. Thanks.
Thanks, Renske. Is it working? Yes. Okay, I want to talk about which dispersal-related traits and why it's important to choose the right trait. So dispersal, we know that dispersal is a very important process that affects both ecology and evolution, and it has a very an impact at local and large spatial scales, but also in small, like short-term and evolutionary, like long-term temporal scales. And of course, us as biologists, we are interested in dispersal because we can ask a lot of questions about it, like why some species have broader geographical ranges than other ones, something related to extinction risk, about diversification. So of course, we are all very interested in understanding dispersal. However, if you are not strictly a theoretician, defining what dispersal is quite difficult, especially because dispersal is a complex process, which can be, we touched this a little bit yesterday with Dries, that it can be divided in 3 different phases: a departure phase, a transfer phase, and a settlement phase. But how do you measure this? How can you measure this, this movement, if it's a movement? So typically, one solution could be using tracking or radiotelemetry to track individuals. You just mark them, you just follow them, you just can have information of their movements, on their activities. However, this is also costly, and you cannot do it on too many individuals. And actually, I heard from some of my new colleagues that to track little fish, one little of these devices might cost 200 euros. So imagine how much money you have to spend to be able to measure dispersal. So, of course, a solution that is actually quite good is to, okay, let's use proxies of dispersal, dispersal-related traits. But those are many. So which trait? So, of course, all these traits are related to the potential of like they have some relation, they will give you some potential for dispersal for individuals. And I want to take you to tell you one story of how the choice of trait that we do will affect the outcomes of our results in our scientific questions. So I am interested, I am a macroecologist, and I am interested to know why some species have larger distributions in space than other ones. So if you see our friend Mola Mola that is just there on top, she I don't know, is distributed all over the world on the sea. But and also our friend Tito Alba, the common bar owl that is somewhere there, I saw it the other day, is also everywhere in the world. However, there are other species, like the Iberian lynx, only occurs in some places in the Iberian Peninsula, and other friends like our rhino, which is not the same that are on top there, but it's the Sumatra rhino, it lives in some islands only. So why? One typical, well, the most obvious thing that I can think of is that they might differ in their dispersal abilities. And it comes to why dispersal, why it's important. And dispersal is important because it allows individuals to colonize. Well, let me see if I can point. I cannot point anything. Okay. Allows individuals that live, let's say we're talking about corals and reef and fishes, to colonize new habitats. When you arrive to a new habitat, sometimes this habitat is not very optimal, so possibly other conditions. So it also helps you to the populations of individuals that are in this new habitat to locally adapt, and it also brings a couple of rescue effects that are more demographic, so the population don't go extinct, but also can allow you to bring this kind of new genetic variants for local adaptation. Of course, dispersal is not always good, and it can also bring maladaptation because of genetic load, but in general, we kind of agree that dispersal should be good for attaining larger range sizes. But is it so? Looks very obvious, but when you see the data, and I collected information of all the studies that have investigated if dispersal has an effect on range size, and from all the studies, you see that the majority find that dispersal doesn't have an effect on range size. There is a large proportion that actually find it's positive, and there are even some that find that it's actually negative. And then you see these mixed outcomes all along the tree of life. Well, this, of course, not all along the tree of life, because there are groups that have never been investigated, but then you see all these mixed outcomes. So I thought, why? So I thought that maybe it's because we are all measuring and doing the things differently. And of course, all the studies differ in several ways. And I thought, yeah, obviously, they measure different dispersal proxies because this is how it is. And I thought, okay, it's one thing that studies differ, but I was not giving that much thought to it. And I thought, okay, they measure the ranges differently. They, of course, use in different realms, different geographical regions. They have different clades. All these things should play a role. But I was not really paying attention too much to these dispersal proxies. When I tried to analyze if all these moderators I call affect the relationship between dispersal and range size, I found out that actually dispersal was always a very important trait explaining whether dispersal will have a positive effect or not on range size. So then I thought, okay, and many other moderators, they were not really that important. Of course, then we found some other things, like whether you are a plant, terrestrial plant, if you are a marine animal or a terrestrial animal. But dispersal proxy was always an important factor. So here I just show you a table of all the different organisms that dispersal and range have been studied on, and different traits that they have been investigated. And then you see the different colors show the different relationships. That green is a positive relationship, the yellowish is neutral, and the red one is negative. And then you see a lot of variation between organisms and within the same species, depending on your dispersal proxy. So I could also do a meta-analysis and investigate each trait independently and see what is actually the overall effect on range size. So I wanted to just give some examples where the traits were good or bad trait for investigating this dispersal range-side relationship. So are big muscles important for dispersal and for attaining larger ranges? I put this insect with big legs, but actually the study was on dragonflies, I think. So you will think having big muscles will help you to move forward and faster and longer, but actually having big muscles has a negative effect on the relationship. The relationship is less strong between dispersal and range size. But then I thought, okay, what about having a big body sizes? Then you see different outcomes depending on the system. Like for insects, there is a positive effect of dispersal on range size. Well, there on top. For amphibians, it's also a good, it's also affect positively the relationship. There is a little bit more variation. And for mollusks as well. But for birds, for mammals, and for reptiles, you see that there is not doesn't affect, there is no relationship. And of course, there is a lot of variation, especially on mammals. What about flying? So for insects, seems that any metric that is related to the wing has a positive effect on range size. And also for birds, like hand-wing index or aspect ratio. But for bats, well, the mean is somewhere in the negative side, but you see this humongous variation. So I don't really know what is happening with the bats. So here is the overall table that we have with all the species and all the traits. And then you see this large variation and this mixed also outcome. So there are some traits that will relate more to range size, and then I will think maybe those are more related to dispersal than other ones. So just as a summary, then that means that depending on the trait you use, you will find out you will have a different story. So if you choose a trait or another one, you might have a completely different outcome. It's a positive dispersal has a positive effect on range size or not. But overall, when I include all this information together, we see that overall dispersal do have a positive effect on range size. But of course, it's difficult to just kind of to find it. So here is just some take-home messages from this, is that we saw that all the outcomes are mixed, but within one species even, in all different realms. But we also saw that in general, there is a positive relationship between dispersal and range size. What is most importantly is that the choice of the dispersal proxy is crucial, and then you will completely find a different, you will have a different outcome in your study. Yeah, with that I want to finish. Thanks a lot. And this was a project that I developed in my previous postdoc in IDIF, yeah, together with Renske. So yeah, I'm happy to receive questions. Yeah.
Thanks very much. There's a lot of time for questions. Who wants to start?
Range sizes are determined by dispersal, but also by other aspects. How do you think that body size, how do you see the role of body size here? Is this purely an effect on the spread or also on endurance, for instance?
Yeah, the body size is a tricky trait, and this is what we have found out during this workshop as well. Because for, maybe I can go back. Like for instance, for fish, body size can relate a little bit with the mobility as an adult, but it also correlates or has some correlation with fecundity. And there is also kind of a syndrome, like bigger fish have more eggs, but the smaller, and the smaller eggs also have more chances to disperse passively. So it's a whole that body size affects several other things as well that end up affecting dispersal. For plants, then we have things like plant height that can be like a body size that will affect how high the seeds can just fall and affect the distance from the parent. But you also have seed size, this kind of thing that will affect not only if they can be spread passively, if they are abiotically dispersed, but then you will see possibly in the next talk that you are maybe a large seed, you might be, and fleshy, I suppose, you will be eaten by like bigger animals and then be dispersed more. So I think the body size is a very key trait, but doesn't need to be directly linked to dispersal, but affects too many things that at the end affect dispersal. Yeah.
It's an interesting discussion because I was also thinking about these kind of big rules and Bergmann's rules or inverse Bergmann rules in insects, where you see that body size is actually also evolving and determining the range sizes or where individuals or where certain species are in the world. So that's why I was asking how you can separate between the effect of dispersal and, let's say, more thermoregulation, starvation resistance, and these things.
I think all those things cover it possibly, and yeah.
So we have a question from the chat. What about seed storage physiology, short-lived versus long-lived?
Seed storage, what was the other thing? Physiology. Okay. That was not really a trait that was considered for this question particularly, but we have discussed about it because that will, and possibly that will also apply for other organisms that will affect the part of endurance, possibly like how long you can just live or being spread. The longer time you can just kind of stay without food or have resources, maybe you are more able to arrive and germinate. Of course, I am not a plant expert, but yeah. But has there been considered in this question? Has never been considered for this question about dispersal and range size.
Thank you, Adriana, for a presentation. I was wondering if you separated for vascular plants abiotic from biotic dispersed ones, because if you think, for example, in fruit size or seed size, the largest one would be dispersed by the largest frugivores. Therefore, would have a large range of distribution, but also in the other side, the smaller seeds from the abiotic dispersed fruits would also reach the large range of distribution. So, did you separate them in a way?
That is totally right. Well, I didn't did this study with like raw data, so it was based on the studies that already did this, so based on how they classify the things. But you are right that, for instance, for plants here, we have diaspore size, assuming that the small diaspores have larger dispersal capacities than large seeds. But if I remember correctly, it was basically for things that are dispersed by wind. But of course, we also have dispersal syndrome, and there are studies that they separate being abiotically dispersed, biotically dispersed, and even they could separate being dispersed by birds, mammals, and different things. And I have seen in this literature that they might consider what has a larger dispersion than the other one in a different way. So some people might think that by wind, if you are dispersed by wind, and possibly you might have a larger dispersal capacity than being dispersed by an animal, but it depends on the animal, because maybe you have very big fly-volant animals, and actually the dispersal potential is larger. So this is something I hope you are also going to help to solve during this week. Because I think with the plants, it's a little bit complex, especially with these syndromes. What is more dispersive than the other one? It might be also context dependent.
There was then I continue.
So thank you for your presentation. Yeah, thank you for your presentation. I had a question about the mycorrhiza category you have. I'm sure we'll learn more about this in the next presentation. But I was wondering if you combined like all mycorrhiza, so those that produce fruiting bodies, and also so the ectomycorrhiza and also the arbuscular mycorrhiza.
This was only one study, and I think they combined both. I will have to double check. But the only, let me see if I have here, wait a second. Because here I have all the traits that actually people use. Where is the mycorrhiza? I know they only use spore volume as the only trait for dispersal. And they didn't find a relationship with grain size. Yeah, he will, yeah. But actually, when I just do the meta-analysis, it seems that it's a little bit positive. Possibly it's because they don't have a very large sample size. Yeah.
I was wondering, so at the beginning of the talk, you explained how dispersal has the 3 phases, right? So I wondered if you found whether one of the 3 phases is more important for successful, if there's more of a strong relationship for colonization, for example.
Well, we try one point to think about the traits and how to classify them in different stages and which ones could be more important. But if it turned out to be very complicated to point which trait will affect what. But today during the workshop, since that we managed to do a classification, but I guess also depends on the organism which thing is going to be more important. For some things, actually, the part that is like the moving thing might be more important, maybe for organisms that are sessile, and then actually they need this transfer phase, and possibly this transfer phase is more important than other ones. But of course, I cannot really say that for sure that this is the way, but yeah.
Thanks.
Yeah, I think the problem was that many of these traits may be involved in several stages, right? So to fix them to one stage is quite difficult.
Yeah, especially life history traits turn out.
Any final questions? Online, anyone? Okay, thanks, Adriana.
Thanks a lot. I'm taking my, wait.
So we're gonna move on to our next speaker, Caroline. And she's gonna tell us more about abiotic and biotic dispersal in plants.
Is it on? Yeah. Hi everyone, thanks everyone for being here, also online. So it's really a real pleasure to be here with all of you. And then today I'm going to talk about some of the main biotic and abiotic mechanisms of dispersal in terrestrial seed plants. So plants are some of the most successful organisms on Earth. They have managed to establish in some places that have very extreme conditions, like low temperatures or high altitudes, but also some desertic places, areas, biomes that are seasonally flooded, for example. They also have strategies to cope with this flooding. And also many tropical forests, for example, where they reach high levels of diversity. But these organisms, they are sessile, so they do not move after they establish. So how they can disperse and how they can establish in so many different environments. So this is what I'm going to talk a bit today. And one thing that is particularly of our interest here is to discuss how different species and organisms have different distribution ranges. So for example, I work a lot with palm trees, and here we have two different examples of two examples of palm species that have they vary a lot in the distribution range. So for example, we have it here, the Mauritia flexosa, that is the Buriti palm, which has a very large distribution range in the South America. And also this palm that has very recently been described by researchers, Brazilian researchers, that is Ciagrus amicorum, which is found in a very restricted area in Brazil, in the Bahia state. So what drives this variation in the range of plants? So one thing that we have also first to keep in mind is that some species, they can only grow under very specific environmental conditions, while some are less limited by the environment. But it's not only that, because species, they also have different dispersal abilities. And this influences the dispersal distance and how far from the mother tree the seeds can reach new areas. So it influences a lot the potential for colonizing and reaching new areas. So these flowering plants or seed plants, they have evolved mechanisms for allowing the dispersal of their diaspores. And the seeds and fruits, they have traits that facilitate dispersal and reaching of suitable locations for seedling establishment. And what I work with is how these propagules or diaspores, they have traits that are adapted to allow for seed movement and colonization of new areas. And there are two main mechanisms of dispersal in plants. One is the abiotic dispersal, and the other one is biotic dispersal. So by abiotic dispersal, we mean that the seeds, they move through by abiotic vectors, such as wind, water, ballistic, or by gravity. While biotic dispersal is driven by species interaction. So animals that interact with plants, seeds, and fruits, and they move seeds around. So here, for example, we can have a look at some of the traits that have evolved that allow for seed movement. So the fruits that are dispersed, fruits and seeds that are dispersed by wind, for example, they have these wings, and they take advantage of the wind to move. Some have explosive mechanisms, so when the fruits dry up, the seeds are released, and they're not dispersed usually very long distance, but they use this strategy. And we also have the water dispersed plants, which they can float. For example, the coconut, and also other palms or the lotus flowers, and then they release their seeds in water, and it disperses by water. And many of them, of the water dispersed, are also other fruits. They can be first dispersed by gravity, just when they fall from the trees, from the parent trees. And some of these, for example, here are some of the structures that we see in fruits and seeds that are adapted to disperse by wind. And you see that they have very different mechanisms for it and very different shapes. And all these traits, they will affect the rate of descent of seeds and how far they can go from the parent tree. But in the tropics where I work, the large majority of plants are dispersed by animals. So that's what I'm going to talk today mostly. So what we see here, for example, in these pictures, are animals that are interacting with fruits. And these interactions, they will allow for movement of seeds. But the fruits and seeds, they have dispersed by animals, they have very different traits. And that's what I'm going to talk today. So how can we link fruit traits to frugivores to understand how these adaptations have evolved? So for example, here we have different groups of animals. We have rodents and psittacids or bats or many bird species, ants, also extinct megafauna fish and ants. So each one of those fruits that are usually interacting with this particular group of animals, they have traits that have evolved specifically to attract these animals. So for example, fruits that are dispersed by bats, they have the seeds that are embedded in the pulp, so that when they eat the fruits, they end up dispersing the seeds. Or birds, they will eat entire fruits, and the seeds are very well protected and they can resist the digestive process, and they are then eliminated, usually, for example, by regurgitation. Others, they don't have much of pulp or flesh pulp, but they invest a lot on seed structures. And those seeds are main foods for example, granivores. In ants, fruits dispersed by ants, for example, they have a structure around the seed that attracts seeds, that attract ants, and they disperse the seeds that we call eliosomes. Or fruits that are dispersed by fish that they colonize areas along streams, and when the fruits fall, they attract the attention of the fish, which then disperse the seeds. And we also have some mechanisms that have evolved to be dispersed by animals without them having to ingest the fruits. For example, these kind of structures that attach to the animal's fur. But the effects of these species interactions is not only on dispersal itself, not only on the movement, because they will affect the entire life cycle of plants. So here we can see, for example, that the animals, they can interact with fruits. They will either predate the fruits or disperse the seeds. And then those seeds, they can germinate and become adult individuals that will again restart the cycle. So these type of interactions have important effects for colonization of new areas and gene flow, and also it will drive the spatial and genetic structure of plant populations. But one thing that we often don't take into account is that when there are many ways that those animals can interact with fruits. So this is what I'm particularly interested on, looking at different types of feeding interactions. So for example, we have fruit eating, pulp eating, and seed eating. So while many fruits are ingested entirely by the animals, there are some other types of interactions, and this also has impacts on dispersal and also on plant establishment. So when we think about fruit eating, that's, for example, what Henske was talking mostly yesterday. The animals, they ingest entire fruits, so they disperse seeds endozoorically. And because many of those animals, they have a large body size and they disperse at long distance, these animals, they can disperse seeds at very long distance. In pulp eating, the animals, they only feed on the pulp. So often, they are considered cheaters or neutral in the system because because most of the research shows that the seeds remain near fruit sources. But this is something that we've been questioning in the last year. So they actually can move seeds far away even if they're not ingesting the fruit entirely, they can carry the fruits to long distance. And also seed eaters, which have been considered for a long time just as seed predators, because they feed primarily on seeds, and they consider low quality seed dispersers because they disperse seeds at a short distance. We've been showing that actually this is not exactly true in some cases, because they can disperse seeds far away. So what I've done is to look at how these different behaviors, also morphological, physiological traits of animals can affect the interaction outcome, and how they form antagonisms where the plant would not benefit from the consumption, but how can they form mutualisms. Another thing that is important to take into account is that quantifying the full effects, all the effects of frugivores on plants is a very tough task. It's not easy because besides the effects of frugivores go beyond seed movement. So they affect seed viability, where seeds are deposited, and they have an important impact on the germination and seedling emergency. So for us to understand how biotic dispersal drives the range of plant species, we have to incorporate all the effects of these interactions, not only on dispersal, but on deposition and seedling establishment. And it is, again, a difficult task because the same animal can have different impacts on different plant species. So we need to integrate this interspecific and interspecific variation of frugivore effects to understand the effects of frugivores on plant community and plant dispersal. But again, how can we do that? So, for example, for measuring all these stages of demographic process, we have to collect palm fruits, but collect seeds and fruits and assess context-dependent effects. For example, how many other fruits there are that frugivores can access, can consume. You also can use, for example, camera traps that will allow for looking which animals interact with which plants, but we also have to assess how seeds establish after the interactions take place. And for that, we also have developed some toolboxes that can help understand how these different demographic processes can be affected by animals. But as Adriana mentioned in her talk, this is very difficult because it's often expensive, and tracking seeds is something that has been done, but it's extremely expensive and also very difficult to really track them. And what I've been working on is mainly with the American palm. So there are around 550 species in the Americas, and they're very important structural elements of these neotropical biomes. They're also a very good study model group because they widespread, they have large variation of fruit traits, and they mainly dispersed by animals. And more than that, they are also provide very important ecosystem service for people because palms are used in many, many different ways. So it's important to understand how animals can impact their colonization, species colonization and persistence in the Americas. So for that, I took advantage of all the literature that we have on palm frugivore interactions to compile. Okay, so we have many palm species, but what are their traits and what are the species that are interacting with the palms? So for example, here are what we got. So we got the information for 175 palm species and almost 500 frugivore species or more, I don't know, that interact with these palms. And we can see here in this network also that some palm species that are shown green, they are dispersed, dispersed by many different groups of animals, but some of them have specific groups of dispersers that are interacting. For example, this one that is mainly eaten by fish, or some that are also eaten by birds and mammals at the same time. But what are the actual effects of those interactions on the full life cycle of palms? So here what we did was to look at how different frugivore traits in each different type of interaction, how it affects the proportion of positive outcomes. So what we did was to compile all the interactions and to classify it as a positive or negative outcome. And what we can see here is that for fruit eating, which is the type of interaction where animals consume entire fruits, most interactions, they result in positive outcomes. And the same is for pulp eating. So what we'll look at if the fruit handling ability of pulp eaters would affect the outcome of interaction. But we see that actually they have very positive effects on palm seeds and palm establishment. But when we look at seed eaters, indeed, we have a lower proportion of positive outcomes, even for those that cache seeds and also for animals with different handling abilities, fruit handling abilities. And when we look at all the species that interact, that we could aggregate studies to calculate the proportion of positive outcomes, we see that there is a lot of variation. So these species interactions, they will affect differently the palms. So what we see here, those that are fully mutualists, they are mainly fruit eaters. So indeed, fruit eating, it seems to favor the dispersal and establishment of palm trees. And we can see there that many of them, they have large body size. So even for the birds, those birds are large-bodied birds. But some of the largest animal dispersers of palms, for example, the tapirs, they are not fully mutualistic. So that shows that they can also have a negative impact on some plants. And the ones that are mostly antagonistic, but not fully antagonistic, like peccaries and some tortoises or squirrels, they are mainly seed eaters. So we do see that there is some pattern emerging from the type of interaction that they establish with palms. But still, we see that because most of them are not fully mutualistic, so they have different impacts on palms. They can both be, interactions can result both in positive and negative outcomes. So what drives this variation in outcomes? When we look at for the frugivorous species for which we could find quantitative data on the specific outcome with different palm trees, we can see for the red-humped agouti, for example, that's the Proctaleporina, which is mainly seed eater. The outcome varies a lot. So for some palm species, they can facilitate the dispersal and establishment of almost 70% of the seeds. But for some of them, they have no positive outcome. And the same happens for the hyacinth macaw and the lowland tapir. But what drives this interspecific variation on the outcome? What we saw is that fruit traits. So, for example, when we look at the effects of the agouti on palm dispersal, we can see that for this palm tree that have small fruits with a very thin endocarp, so the seed is not very well protected, the agoutis, they kill all the seeds. But when they disperse seeds that have stony endocarps, they can actually benefit dispersal and establishment of a large proportion of seeds. The same when we look at the macaw, so it has very different effects according to the traits. And for the tapir, it's interesting to see as well, because for the same palm species, they can have different effects, as we can see here. So some, for the Attalea maripa, for example, can benefit almost to a full extent from the interaction, but not in every context. So there are context-dependent factors that are also driving these outcomes. Okay, so there are, what we found therefore that indeed there are some of these traits that link the frugivores, that link traits to frugivores. But what we need to understand the full consequence of this biotic dispersal, we need to integrate all this interspecific variation and also the traits of animals and the type of interactions to fully understand the effects of dispersal on plants. So this is not simple because they have very different effects on each demographic stage of plants. So here we summarized the main sources of behavioral variation and also trait variation, so of how they can affect the different demographic process according to the way they interact with seeds. Why is it important to understand how the way the animals interact with fruits? Well, we know that the loss of megafauna, for example, has led to the shortening of seed dispersal distance, as we can see here in this paper of Matthias, where we see that seeds were dispersed very far away by the megafauna, while nowadays the contemporary dispersers, they can disperse seeds at lower distance. Yet, no, sorry, and we still have to consider the potential for dispersal distance because of, for example, climate change, because plants that are dispersed biotically, they would depend, they depend on the animals for dispersal, and we need to assess how these impacts will, the climate change, for example, will affect the animals with different traits and what are the consequences for dispersal services. But what we need to take into account is that although the loss of large mammals might disrupt fruit-eating interactions, there are also the seed eaters and the pulp eaters that can actually take seeds very far away from the source. So we have some examples here of studies that evaluated dispersal distance by, for example, parrots or fish, or also the agoutis that are mainly seed eaters, that they can actually disperse seeds very, very far away. So what we found also is that ectozoochory, that is the interaction in which the animal does not ingest the entire fruit, so it carries the fruit or the seed on their mouth or beak, but not endozoochorically. They can disperse seeds at large distance, like we can see here. In this graph, for example, we have the mean dispersal distance and the maximum dispersal distance. And we see that even though fruit eaters that are in blue, they indeed have dispersed seeds at the largest distance, the seed eaters and the pulp eaters can also move seeds. Why this is important also for understanding, for example, the effects of interactions of biotic dispersal on ecosystem dynamics. So we also did this work where we showed that even though the largest extant megafauna in the neotropics, they disperse seeds, large seeds that are linked to high carbon storage. Agoutis, for example, by ectozoochory, they can benefit seedling establishment and dispersal of species that have an even higher seed mass than the extant megafauna that we have, and also with high carbon storage. So these interactions with pulp and seed eaters, they are less constrained by size matching rules, so they might be some of the main agents for large dispersal in the faunated areas. And there is an important impact on the potential and the contribution to carbon storage. So just to conclude, what we've seen here is that plants have evolved different mechanisms for dispersal, and that particularly in the biotic interactions, the frugivores, they play important roles on different demographic processes of plants. So they have an impact in shaping complex plant dynamics in ecological communities. And I couldn't stress more the need for taking interspecific and intraspecific variation of animals' roles into account to understand how this has evolved and how these interactions have shaped also fruit traits and how plants can take advantage of these interactions. But for that, we also need to integrate data on movement ecology of frugivores so that we can know how far the seeds can go, where they can be deposited, and how this will impact the plant establishment. And yeah, just thanks to everyone and for the invitation, and also to everyone that helped me somehow to do my work. Thanks.
Great, thanks a lot.
Thank you.
Questions?
I just have a very naive question, possibly. Okay. When you were talking about the antagonism and the mutualism, how do you measure it? Like, something with the morphology? How do you know which animal is actually predating or how much predating? How do you just put them in this kind of gradient?
Okay, so most of the studies, they they assess the outcome of the interaction in those ways, for example. So for example, after one fruit eater eats a fruit, then people collect the seeds from they can find fecal samples on the forest, for example, and they can assess the viability of the seed by experiments. So then they will conclude that the dispersal was positive, the effect of the frugivore was positive because the seed survives the retention time and digestive process. But in some cases, they can also place seeds in experiments to see if seeds germinate after that, or if they establish, and what are the effects if seeds that are not ingested by animals, they have a higher germination success than the ones that are. So from this, we can know how many seeds have benefited from the interaction, and then we can calculate the proportion of seeds that indeed survived the interaction. And so then by aggregating data on this, we could see how this proportion, the proportion of positive outcomes. So here we only found studies that found the effects of those frugivores are fully positive. So that's how we knew where in the continuum the frugivores are.
Very nice talk, thank you. So many of these animals also eat fungi. And I wonder if is it what you would expect, that there's competition between the plants and fungi for dispersers, or has anybody looked at that?
Okay, thanks for the question. So you mean how much of their diet is made of different items? So yeah, this probably has an impact on the outcome, because some animals that are they are not the quantity of fruits in their diet is low. They often have they have a negative impact on seeds, because well, we have a hypothesis for these cases that the seeds didn't evolve in response to consumption by some non-frugivore animals, for example, so it can affect negatively the seed. But I didn't look how much of their consumption on fruits and seeds have impacted. I think I actually looked at it, but we didn't find a pattern, a clear pattern. So it depends on the seeds, yeah, and how they interact with particular traits or particular species, I'd say.
Okay, one.
Okay. Thank you for your talk. Is there any kind of morphological, physiological, or any kind of trait we could use as a proxy for this kind of antagonism relationship?
Well, yeah, that's tough, because I think, well, what we can see here, for example, is that the species that are less mutualistic, they still have positive effects on plants, but that most of the interactions result in negative outcomes. They are dependent on the seeds. So I think that there are possibly some traits that those species share, or food preferences, or that have evolved and that they do not promote much benefit for the plants because they eat the seeds. But we didn't find a clear relationship of body size or something like that. And the difficult thing is that these outcomes, they very specific, species specific. So as I've shown in here, even the same the same palm species can have different effects depending on what the animals are doing, also on other food that they're relying. So they might eat more seeds if there is less food, so then it's negative. So I think that this can affect, and possibly other traits as well, I'm pretty sure.
And what about the other side, the animals?
The mutualistic.
Yeah.
Well, yeah, those are these, again here, the mostly mutualistic or fully mutualistic animals, their behavior is ingest entire fruits. They do not feed on seeds exactly. And the fruits have evolved adaptations for this type of interaction. So they have sometimes long retention times, but the seeds can still survive. So what we look at for fruit eaters mainly in this paper was, where is it? No, sorry. Was whether regurgitation or defecation of seeds would affect, because in defecation, the seed goes through the entire animal's gut, but it seems that those seeds are adapted to resist this processing. Yeah.
Thank you.
You're welcome.
Who was next? One here.
Yeah, so I had a question as well about the antagonist mutualist spectrum. Because a proportion of positive outcomes of 0.1 might actually be a very good thing for a tree or a plant that produces a lot of seeds, right?
Yeah. Oh, you mean that it would be a good...
So it can be a mutualism if the plant produces a lot of seeds, then a proportion of positive outcome of 0.1 might still be a lot of seeds compared to...
Yeah, indeed.
Yeah. So is it fair to call it an antagonism then?
Well, we're actually not calling them antagonistic, but mostly antagonistic, exactly because we're taking into account that they have positive effects on seeds, but it's true. And some of the seeds, for example, dispersed by squirrels or spino rats, they actually have, they produce many, many fruits. So they still benefit from the interactions with those animals, even though they're mostly antagonistic. And that's what we've been seeing in many studies also, how, for example, again, among the plants that produce many seeds, they can satiate animals. So they can, for example, scatter hoarders that they cache seeds for later consumption when food is scarce. They might depend on this 10% of positive effects. Yeah.
So there's actually one question and 2 comments online.
Okay.
So first the question, thanks for this great talk. One naive question, do fish dispersed species in neotropical palms grow in seasonally inundated areas? And how do they reach final establishment inland? Yeah,
that is a really great question. And actually I was just last month, I don't know where, in Pantanal, in Brazil, collecting palm fruits. And this palm here, that is a Bactris from the genus Bactris. We still don't know which species it is. We are collecting some more trait data to know. This area here, it's now it's in the dry season, but they are flooded. So fish do disperse palm seeds, but we still don't know exactly how the dynamics of these seasonally flooded places interfere on the establishment. But this is a very interesting question that it would be great and probably very difficult to answer.
And then there's a third comment as well. Yeah, it's concerning some of the other questions. So someone says that seed surface fungus may have a negative impact on seed dispersal because the animals don't like the taste.
Yeah, that's pretty cool. And I'm here, one of these here that I'm showing, for example, is not on fungus, but I'm also on fungi or pathogens, but I'm also interested in looking what happens with the seeds that are not immediately dispersed or consumed, because then they concentrate in near parent plants, and because of a higher density, they are subjected to higher predation, and so this affects how many seeds will be dispersed in the end. Yeah, that's nice.
And then there was a comment about quantifying the antagonism. So someone says seed predation out of the total seed crop might be a factor to quantify antagonism in plant-animal interactions.
Yeah, indeed.
And then finally, someone suggests that Dr. Sandra Correa, I hope has done a lot of work on ichthyochory, so the fish seed dispersal. So.
Oh, that's cool. It's good to know.
And now there's another question. How important do you think intraspecific variation in conspecific seed dispersers is for seed dispersal outcomes? How important? Yeah, okay.
Yeah, I think, sorry, can you read again? Intraspecific variation of conspecific?
Intraspecific variation in conspecific seed dispersers. How important is it for the dispersal of the seeds?
Well,
the outcome.
I suppose it's important that they have a positive outcomes, and I think what will play a role in this intraspecific variation is mostly context-dependent effect. So how much food there is, and how far from periods of scarcity the consumption is taking place. Yeah. I hope I answered it, otherwise I can answer it later on.
I still have time?
Yeah. Oh. Yeah, we were a bit early.
Okay. I was not expecting to ask a question now. I had a question about the fact, can you give me a little bit more explanation about the fruiting, let's say, timing of these palms? Do they fruit at the same time, same species? Or is this all randomly throughout the season?
Yeah, most palm trees, they produce fruits every year. But for example, I did work during my PhD with an Astrocarium palm tree in French Guiana, which is a masting species actually. It produces fruits every 4 or 5 years.
And but within the same year, is it all simultaneously? So do you have a kind of, let's say, all the like our cherry trees, they're all blossoming in beginning of April, somewhere April. Is it the same for palms? Do they have very restricted fruiting periods?
There is a lot of variation. Yeah, there in Pantanal again, there was one palm that all adult individuals were flowering, and they do that every year, for example. But some, we had the fruits that were just being produced and mature fruits, and it takes quite some time for fruits to mature in palms, in some palms, so there is some variation.
Yeah, because I was thinking, because if this bird feeds on a certain palm tree and there's only the same species which is fruiting everywhere, then I would expect them to fly to another palm tree to eat some over there and another palm tree over there. And then the seeds would eventually drop below the palm trees, which is maybe at first glance they would germinate, but in the end, I think the Janssen-Cornell effect, that's what I understood, is that they would actually not be so benefited because they end up somewhere in the neighborhood of the same species.
Yeah,
I was just wondering whether that was
no, indeed that could happen, for sure. And I suppose that this is more common with the terrestrial animals, also the ones with that are not that don't have very large body size, so that they move to shorter distance, and then they can move seeds to below another conspecific palm tree. So it's just, this can happen, but then the seeds won't benefit most likely, because
or they will compete with other species. I was just wondering how it works, because if it's interesting to understand the behavior of the frugivores as well. If they would drop the seeds somewhere at places that are quite open and very light rich. I don't know anything about these palms. I can imagine that the fruits, the seedlings could have a really benefit.
Yeah.
But if they would end up in the shadow, maybe then
definitely. And I didn't work exactly with that, but reading all these papers, the literature on palm frugivore interactions, there are some cases where seeds, what we call directed dispersal, so that sometimes they move seeds to the same place, and then there is the seeds are viable, but there are just so many that it's as if the seeds were near the parent tree and it does not benefit, but in some cases they do.
Yeah.
Yeah, so there is a lot of variation. It's just two really many interactions and.
Yeah, but it's interesting because the mutualism that you have then maybe in your graph might eventually not be a mutualism in some cases. Maybe I don't know. I mean,
because you think like we could be measuring seed viability, but not the effect on seedling establishment.
I don't know what
Definitely.
the end point is where they measure. I mean, success of the dispersal event. Did they effective, or did you have measures about where the seed ends up, whether the seed germinated, whether the seedlings have survived, and so on?
Yeah, so that's what we did. We compiled it, but most studies, unfortunately, they do not do all the demographic process. So we just tried to aggregate all these outcomes to understand a little better the picture of the role of those animals.
Ah, but nice work.
Thanks.
All right, thanks a lot, everyone, and thanks a lot for your talk and loads of questions.
Thanks.
We'll have a break now, I think, right? Until 5:30, and then we'll be back. So see you in 20 minutes.
Ready to start? Is this working? Hello? Oh, okay, I see.
All right. Welcome back, everyone. We're going to continue with the next talk, going across spatial skills in fungi with Bala.
Thank you. Okay, I am very happy to be here talking to you all in the colloquium. I'm very happy to be here in the symposium as well, representing fungi in the world of dispersal. It's a little complicated, which I will get into shortly, but I wanted to mention that a lot of the ideas and concepts I'm going to be talking about today briefly are in a paper that colleagues and I just published last month in annual reviews called Fungal Dispersal Across Spatial Scales. It was the result of a sabbatical year that I spent in the lab of Matthias Rillig at the Freie Universität in Berlin. And I just want to acknowledge him and his group members, Carlos and India, who also greatly contributed to this work. Lots of conversations, some in the beer garden. It was a really wonderful time to spend in another group thinking and learning about fungi. They have a very vibrant group there. Okay, so when we think about fungi, often this is what comes to mind. So primarily mushrooms, right? Maybe sometimes you think of the mold that grows on your bread in your kitchen, but usually we think of mushrooms. And it's important to keep in mind that mushrooms are really just the showy fruiting bodies of fungi, and we refer to those fungi that form visible fruiting bodies as macrofungi. But below ground, this is what fungi look like. And most fungi spend their entire lives in simply this form, below ground network of hyphae called mycelium. And they are often just these hollow tubes. And they occur like this in soil, but also lots of other locations as well. I wanted to bring up specifically soil fungi because we just this past Monday passed World Soil Day, which is December 5th. And there's a lot of excellent educational information out about World Soil Day, which happened about 10 years ago. It was the first World Soil Day put out by FAO at the UN. And I like to share this statistic that if you think about soil, often there are more organisms in 1 tablespoon of soil than there are humans on Earth. And so I think it helps us think a little bit, as we've been hearing these amazing dispersal ecology talks about macroorganisms, it helps us think a little bit and maybe shrink down to the scale at which some of us work and frankly where the majority of life on Earth exists. Okay, but back to fungi specifically. We estimate that there are between 3 and 13 million fungal species on Earth. So I'm very happy to be here representing fungi, but it feels a little daunting. I'm certainly not an expert in all fungi. We wouldn't ask somebody who works on insects to represent birds, but sometimes that happens in the fungal world. That's fine. So those fungi that form above ground fruiting bodies or mushrooms, they represent about 6,000 species. So we have been studying dispersal of macrofungi, those 6,000 species, for quite a long time, over 100 years. So my colleagues have been studying dispersal in macrofungi quite a lot, but we know very little about the dispersal of the other fungi, which are the majority of fungal species on Earth. Now, what do fungi do? Fungi can be saprotrophs, so they can be responsible for breaking down organic matter in the litter layer of forests. They can be pathogens This is a corn smut that is a fungal pathogen infecting a crop. And they can also be symbionts. So they are fungi living inside, in this case of this diagram, inside plant tissue, inside leaves, inside roots, inside stems, asymptomatically. So they're not forming disease, that's just where they live. And I want to emphasize that fungi exist in terrestrial ecosystems. They also exist in aquatic ecosystems. There's a whole world of marine fungi that we're just at the tip of the iceberg in understanding their diversity and function. And then they can be amphibious. This is a picture of many recently cultured marine fungi. So there's a huge diversity of fungi in terrestrial aquatic environments, and there are also fungi that are found in both locations, so they're amphibious. And it's not necessarily that in marine environments they're just there kind of dispersing or they're temporarily. There's evidence that marine fungi live and are physiologically active and important in those ecosystems. Okay, now we're here to talk about dispersal. And if we're thinking about really the beginning of microbial dispersal, yesterday we heard Katja talk about the Bos-Becking hypothesis. So this is often where we start with microbial dispersal. And the Bos-Becking hypothesis states that everything is everywhere, but the environment selects. And so this dominant hypothesis has come into the fungal world as well, but it gets a little challenging for the fungi that don't form above ground structures. So, for example, there's some fungi that live entirely below ground. So does this hypothesis apply to those fungi as well? We don't know. Maybe, maybe not. And does it even make sense to try to apply the Bos-Becking hypothesis to fungi? And really the question is, are fungi even microbes? And I guess my answer to that is sometimes. So a microbe is, at the core, an organism that requires a microscope to see it. But fungi can actually be quite large. So the largest organism on Earth is a fungus. This is Armillaria ostoi over here, sometimes referred to as the humongous fungus. It's a fungus that exists over about 600 hectares. It's a single genetically identical isolate across this huge area. But they can also be very small. And so these are pictures of tropical leaf endophytes. And so within a single leaf, if you were to isolate the fungi in this leaf, you could find dozens and dozens of distinct fungal species in a single leaf. And often those fungal individuals have been shown to be roughly 2 millimeters square in size. So if you're thinking about how to define individuals, it gets a little complicated. And it's mainly because fungi are modular organisms, as opposed to unitary organisms like humans or some plants. So they defy this unique and unambiguous definition of what an individual is. These are some issues we've been talking about quite a lot this week in our workshop. And it also brings up concepts like the genet. So genets are individuals derived from a single cell or a single spore. And that's compared to a ramete, which can be clonal and really united through their physiology. Okay, so if we're trying to understand fungal dispersal, or even movement for that matter, sometimes I fall back on this definition of dispersal. Dispersal is any movement of individuals or propagules that has potential consequences for gene flow across space. And I've just underlined a few of the concepts that make mycologists uncomfortable. So how do you talk about individuals? Well, how do we talk about propagules? Spores are propagules, yes, but also hyphal fragments can be propagules. For some species, definitely not all. Gene flow is complicated. I don't have a lot of time to talk about fungal gene flow in this presentation, unfortunately. But I did want to underscore space and what are the spatial scales that are important for fungal individuals or important for fungal movement and dispersal. They can be very small or they can be very large. It's not fixed. So in our paper, we tried to identify some of the major mechanisms of fungal movement and put forth some hypotheses about the relative importance of different fungal movement and dispersal mechanisms, and these are particularly at local scales. So at the smallest spatial scale, mycelial exploration is probably the most important mechanism of movement or dispersal. And then compare that to other mechanisms such as movement through micro or macro arthropods, earthworms are just one example we were looking at, and then also movement of fungal propagules by small mammals. And their relative importance likely varies with spatial scale. Now, those were local scale mechanisms of movement, but then fungi certainly move at landscape or even continent and global scales as well through different mechanisms. And so movement of fungi via birds is likely increasingly more important at large spatial scales. There's marine and freshwater movement, wind dispersal, which I'll talk about in more detail here shortly, and then certainly human movement, which can be relatively more important at larger spatial scales. So there's no data here. These are all just thoughts and ideas that we were putting forward to try to stimulate conversation and potential research in fungal movement ecology. Okay, now why would we care? You all didn't have to explain why anybody would care about your organisms, because look around, everybody cares about these organisms. But I've been looking hard for fungi, I didn't find them. So I thought I would just discuss why we might care about fungal dispersal, why it's important to study fungal dispersal. Well, first, it's very important from a human health perspective. So this is an example of a disease, a human pathogen, coccidioides, that causes the disease valley fever. This is a fungus that lives in soil, and when soil is disturbed, it's released, the fungus is released into the air. Humans can inhale it, and it causes a fairly serious disease. Also, because because the fungus has to be released by disturbance and essentially soil degradation. This disease is much more likely to occur in people that live in disturbed areas, so that includes migrant workers and other marginalized communities. So there's a justice component to understanding fungal dispersal in this aspect. Certainly from a biodiversity conservation perspective, some fungal pathogens have been really wreaking havoc on biodiversity of other organisms. So white nose syndrome is caused by a fungal pathogen that's wiped out almost 90% of bat species in some areas in North America. And then finally, from a food security perspective, fungal pathogens in agriculture are hugely important. This is a picture of farm workers removing banana plants that have been infected by banana wilt. And some of these fungal pathogens threaten our food security around the globe. So understanding how fungi move is really important from an application perspective as well. Okay, I am also interested in fungi from an ecosystem perspective. So in my lab, we study plant symbiotic fungi. And they are fungi that live inside plant roots that are part of the microbiome. And these fungi affect plant health greatly. So not just plant productivity, but also how plants use water, how plants take up nutrients, and then all of the ecosystem services that we derive from plants as well. So in my lab, we study, in particular, mycorrhizal fungi. Those are fungi that form symbiotic associations with plants. You could even say that the reason we have land plants is because of symbiotic fungi who were really central to the land colonization of first plants on Earth. We have fossil evidence of fungi that really haven't changed much from a trait perspective over 460 million years. And plants form these associations with mycorrhizal fungi in a nutritional symbiosis. So the fungi take up nutrients from the soil, nitrogen, phosphorus, and they give it to the plant in exchange for carbon that the plant fixes into sugars through photosynthesis. There have been a lot of other studies showing how mycorrhizal fungi benefit plants in other ways aside from productivity, such as pathogen protection, drought tolerance. They affect plant interactions and have been increasingly shown to be important in carbon sequestration globally. In my lab, we study a particular type of mycorrhizal fungi, arbuscular mycorrhizal fungi. Now, these mycorrhizal fungi don't form any above ground mushrooms. They exist purely below ground, and they reproduce with asexual spores that bud off and live entirely in the soil. Now, from a dispersal perspective, because they exist entirely in the soil, they were thought to be quite dispersal limited. But that was not really jiving with the data that we have that shows similar species, similar groups of species living in disparate locations around the world. So they exhibit very low levels of endemism and cosmopolitan distributions. So we were really coming at this dispersal question primarily because deterministic and niche-based processes were not completely helping us understand community assembly at local scales and biogeography at larger landscape and continent spatial scales. So we were wondering more, like maybe there's something more to their dispersal that we didn't understand. And we conducted a study. It was in urban green roofs where we were really trying to understand what are the best predictors of mycorrhizal fungal abundance in these highly urbanized environments. And so we were trying to understand whether soil properties were the best predictors of mycorrhizal fungal abundance, or maybe whether plant properties were the best predictors of mycorrhizal fungal abundance, which you would expect because they're obligate symbionts. But really what we found, and I want to draw your attention to this, sorry, there's a lot of numbers here. This was the strongest direct effect or the strongest predictor of mycorrhizal fungal abundance was the age of the roof. Nothing that had to do with soil properties, nothing that had to do with plant properties. And these categories were really primarily deterministic, and it got us wondering if maybe there was passive dispersal happening into these green roofs over time. But how are they getting there? How were these fungi that live entirely below ground getting onto the rooftops of skyscrapers, 50 stories up in the middle of Chicago? It was a bit of a conundrum. And that's when we started to realize, wow, we don't know very much about mycorrhizal fungal dispersal. If they are dispersing, how do they exit the rhizosphere? Do all species disperse in a similar manner? We shouldn't expect that, although that had been kind of the prior understanding. And is there a way to potentially predict dispersal using spore traits? And then again, we really wanted to try to fill some of these gaps because dispersal gaps really hinder our ability to understand mycorrhizal fungal community assembly and also their management in ecosystems. They're of great interest to ecosystem management and use in sustainable agriculture. Okay, now what about these spore traits? AM fungi, or buscular mycorrhizal fungi, form spores below ground that vary with respect to a suite of quantifiable traits. They form the largest fungal spores of any fungal group, and so they actually make quite a few different traits that can be quantified, and these traits likely affect their movement, their persistence, their ability to survive in the environment, and they also affect the amount of resources that they need when they're trading with their plant partners. So we did a study to try to understand how prevalent aerial dispersal was in AM fungi. We wanted to know whether we could predict which species disperse aerially using traits, and then we wanted to understand whether there were any temporal patterns in dispersal that could inform these mechanisms. So we conducted month-long aerial sampling, and we assessed the fungal community in the air using metabarcoding, but then we also measured the traits of all the spores that we found in the air. Okay, we found there were tons of AM fungal spores in the air. We measured traits of 47,162 spores. My advisor back in the day always told me to explain the number of spores you counted. And we found them at all months of the year, even winter, and then a fairly high number of different species identified morphologically and with the genetic techniques. We did a heat map of the traits of the spores that we detected in the air, and we could see that most of the spores that we found were on the smaller side, on the smaller range, which is what you would expect. But there was quite a range of traits with respect to their pigmentation and also their ornamentation. Then what we could do is compare the traits of the fungal spores that we found in the air to kind of the variation of traits that you see across this entire group of fungi. So all the AM fungi, the AM fungi are monophyletic, and they're all within the Glomeromycotina. And so we found that aerial spores were indeed smaller with respect to size compared to the fungi across all of the Glomeromycotina. And so this is kind of what you would expect, but it was interesting to us that about a third of described taxa form spores that are within the size range of the spores that we detected in the air. And this indicated to us that aerial dispersal of fungi is likely more common than previously thought. We also found a pretty strong temporal pattern, so that aerial dispersal of these fungi was primarily in the fall, so between August and November. And then we saw lots of species-specific temporal patterns as well. I'm not going to go into all this detail here. When we looked at the communities with the metabarcoding technique, we could then compare what we found to global databases, and we kept identifying species that had been previously detected in agricultural soils. So this led us to wonder, why do we see this big peak in aerial dispersal during these months? So we didn't really see any strong patterns with traits or with meteorological, patterns. And so we started to wonder if maybe this is a time, we know that this is a time in our region where there was quite a lot of agricultural soil disturbance. So then we could get some landscape level plant phenological data and show that plant senescence, in the year that we did our study, plant senescence was in fact temporally cued with when we saw spores in the air. So there was a link between likely soil disturbance at that time and fungal sporulation. So the combination of fungal sporulation and soil disturbance at that time likely led to the peak in aerial dispersal of fungi in the fall. So just some take-home messages from this. We're hoping that traits can help us predict which fungal species are more likely to disperse aerially, and that it's possible that anthropogenic soil disturbance is spreading fungi and promoting disturbance. But it's not just spreading all fungi, it's spreading these disturbance-loving ruderal fungi that are more likely to live in agricultural areas, and that could lead to potential homogenization of mycorrhizal communities across spatial scale and teleconnections that exist globally with soil disturbance. I wanted to throw this up here because we talked a lot about the 3 steps of fungal dispersal, and this is something that folks in the group are thinking about too. I think our 3 steps of fungal dispersal matches very well with fungi. And then next, moving forward, we have a study planned to try to test how generalizable our previous studies' results really are and whether we can predict trait-based aerial dispersal at larger scales. So we've installed dust collectors, passive dust collectors, at 20 sites across the US, including in Alaska, Hawaii, and Puerto Rico. And we'll be looking at the fungi in the air and also spore traits. Oh, maybe I can say a couple more things. If you don't mind. Oops. Okay, some preliminary data from our continent scale study shows that we're measuring spore traits, but we're also measuring spore viability. And so we can collect spores from the dust and we can stain them with a vital stain. And it looks like the spores that are more likely viable have specific traits. And one of the common traits are thick spore walls and complex spore walls. So I think this is another trait that's kind of popping up to us as something that facilitates dispersal. Oh, I think I better wrap it up here. Sorry. So I really really overestimated. Okay, last point. There's a lot we have yet to do in the fungal dispersal world, but I think it's an excellent time to enter the field of fungal movement ecology. It's a time when we have lots of different methodological and systems advances coming together to really help us answer big questions in fungal ecology. So come and join us in the fungal dispersal world. And I'll just stop there and thank my lab collaborators and funders. I especially want to thank the Douglas C. Florin Fellowship that helped bring me here to Leiden.
Thanks a lot. Fascinating. We have 1 minute.
Sorry.
We can take a few more minutes. Are there any burning questions?
No one's burning.
Yeah.
So how important do you think these aerial dispersal is for ecosystems that don't have a lot of soil disturbance?
Oh, good question. And I think that's one thing we're tackling with our new continent scale study, because I think aerial dispersal is, of course, one mechanism, and then we talked about other small mammals and other you know, birds, etc. I think that the dominant dispersal mechanism varies with system. And you know, if you think about a dense forest, I don't think there's a whole lot of wind dispersal happening there. I think it's primarily small mammals. And I have a collaborator, Ryan Stevens, who showed that in a small study in New Hampshire, where I'm from. And then I think disturbances, in areas that lack disturbance, it's potentially other mechanisms as well. But every system has disturbance, you know, whether it's anthropogenic or natural. Small mammals cause disturbance, hurricanes cause disturbance, and so I think it's part of it.
Any other questions? Yeah.
Thanks, Bala. That was really great. I've learned a lot because I actually don't know much about fungi. But you just mentioned hurricanes, and that was what I was thinking. If there is some study, or if you do some research on how extreme climatic events can change the potential for dispersal in these organisms.
Yeah, I think they're fairly understudied. I mean, certainly there's the wind aspect of the hurricane, but I think what's probably the more impactful thing that happens with a hurricane is tree uprooting. And so then, you know, when you see a tree that's been pushed over by a hurricane, all the roots are in the air, all the soil is kind of flipped up, and I think that that's a disturbance event. But there's no data there. It's very data free.
So we have another online question. Have you considered seed surface assisted dispersal of fungi?
Yes, we haven't studied it directly, but there have been other studies that showed this. So there's a very famous in my circle study of a seed and mycorrhizal fungal co-dispersal by a bird. And so I think that's a potential option, even if the fungi are not on the seeds, but I think there's a lot of co-dispersal going on with seeds.
Cool. Cool, cool. Nice. Thanks.
Thank you.
We're going to move on to the last talk. Don't leave. No, it's okay. Thanks for the questions. We're going to move more again to the evolutionary side of dispersal, thinking about hybrid zones with amphibians and with Ben.
Now it's on. Yeah, okay, sure. Well, yeah, thank you. Nice to be here in Life Science. Thank you for inviting me. So yeah, dispersal over evolutionary time. Hybrid zone movement, dispersal over evolutionary time. What do I mean with evolutionary time? A bit of a vague term maybe, but yeah, it's particularly about dispersal happening over many, many generations. So say hundreds or thousands of generations, hundreds of thousands of years. And what do I mean with hybrid zone movement? Well, let me explain that in a bit more detail. So this gray blob is supposed to represent the range of a species. And what often happens in nature is that barriers appear that break up a species range. So that might simply be a zone of unsuitable climate, a mountain range, a river, a desert, whatever. And what happens if that, if those two populations become isolated, if gene flow becomes impossible between those isolated ranges, they start to evolve independently, and just give it enough time, these isolated populations will start to accumulate genomic incompatibilities, which basically just means if you take an individual from one and the other population, you put them together, the longer the time that has that has passed, the less compatible the genomes are. So the less well those genomes would be able to collaborate in a single individual, in a single hybrid individual. Now, barriers disappear all the time as well, just as they appear all the time in nature. And if that happens, then yeah, these populations can move towards one another again, and they can obtain secondary contact. And if they have accumulated sufficient genomic incompatibility, they can remain their genetic integrity. So they are, for all intents and purposes, you could treat them as species at that moment. But if reproductive isolation is not zero, then hybridization could occur, and hybridization occurs in a certain geographical area, and that you would call a hybrid zone. So that is the region where these individuals of different species meet and mate and produce hybrid offspring. Now, that moment of secondary contact is in a way quite random. Obviously, it depends on circumstances, dispersal capabilities of these individual species. But at a certain moment, they will just meet somewhere. And that does not mean that they are then in, say, competitive equilibrium. So if one species has a competitive advantage over the other one, it simply will start to expand at the expense of the other one. And then the hybrid zone that separates their ranges will basically move across the landscape. Now, many hybrid zones are actually very old. Well, very old. Say, for example, in Europe, many hybrid zones, basically all the hybrid zones we have here, simply occurred after post-glacial expansion. So say up to 12,000 years ago or something, range were regressed because it was much, much colder at the time. Since then, glacial conditions alleviated, species started expanding and started meeting up at hybrid zones. So there has been a lot of time available, basically. A lot of generations have been available. So yeah, that means that hybrid zones could potentially have moved large distances. Now, if you look at the classical literature on hybrid zones, then hybrid zone movement is actually considered quite inconsequential. You know, it's acknowledged that it happens upon secondary contact, but it's something that just very quickly they would stabilize. There is actually little empirical evidence of hybrid zone movement over vast distances, fast timescales. But on the other hand, we have been studying hybrid zones in some cases now for decades. And if you just look at those hybrid zones long enough, you can see that they are moving. So if they can move over decades, yeah, why can't they do that over hundreds or thousands of years and potentially cover large ranges? As I said, little empirical evidence, but you know, you need to look for it if you want to detect it, right? And maybe that is where things have been missing out a bit. What is often observed at hybrid zones, I would go as far as saying it's the rule rather than the exception, is that there is introgression at hybrid zones that is geographically asymmetric. So what you see is genes that evolved in one species, or alleles I should say, variants of genes that evolved in one species, are actually distributed in the range of the other species. Particularly, for example, mitochondrial DNA. Maybe simply because people start looking at mitochondrial DNA already, yeah, several decades ago by now. You just really regularly see asymmetric introgression at hybrid zones. But yeah, that's often based on single markers. Now, if you consider that pattern, how could that occur? So kind of like a traditional interpretation of that pattern, I would say, is that positive selection is at play here, so that particular allele of one species simply outperforms the allele of the other species. So what you see in this picture, I'm not sure if this pointer is actually working. No, it's not. So this is the place of the hybrid zone. The red allele has introgressed in the green species, and it kind of starts moving towards the left simply because it outperforms the green allele. Kind of like an alternative explanation that is increasingly invoked in the literature is that hybrid zone movement is at play here. So here's something else happens. It's not the individual gene that is moving. It is the hybrid zone itself that is moving. It kind of moves over the gene. So what happens here was the initial hybrid zone. A red allele introgresses into the green species. The green species expands. So all the offspring, all the colonization is being done by green individuals that have that red allele introgressed. So that also means that as this hybrid zone moves, in the wake of the hybrid zone, you will find the red allele. But yeah, as I said, often based on single markers, and it's actually difficult to distinguish between these two geographically quite distinct processes. However, we can now look at genomic data, genome scale data, many independent markers. And I would argue that that can help you to distinguish between these two scenarios. So perhaps it's best to think about this in the context of a geographical cline. So a geographical cline is basically a transition in space from one thing to the next. So here you see a geographical cline for redness, being red, the frequency of being red. If you are on the left side of this plot, you are completely green, so you are not red. If you move towards the right, you hit the hybrid zone, and individuals overall are more and more red until you hit the core range of the red species, and then you are completely red. So you have that transition from green and red, and that can be based on, say, a genome-wide average, genetic composition. But if you look at individual genes, you can also look at geographical clines. And if you think about the scenario of positive selection, what you would see is that, yeah, these individual clines are being pulled in. That is an ongoing process that at a certain moment simply reaches fixation and you lose the signal, right? If the red allele completely displaces the green allele, you don't see it anymore. It's just all the same. Also, individual alleles can be selected on, and they could potentially move in different directions, so independent of the rest of the genome, right? Now, under hybrid zone movement, you would actually predict a very different pattern. So just alleles that are selectively neutral, for which it doesn't matter if you have the red or the green allele, they could potentially introgress, but they would always introgress in the same direction. They will always be present in the wake of the moving hybrid zone. Also, this is much more temporary stable. Once this pattern is fixed, there is not really a drive to get rid of it, right? It's only susceptible to genetic drift at that moment. So you expect this pattern to be retained for much longer time periods. So this quite distinct pattern from expected under positive selection, I call it a genomic footprint of hybrid zone movement. And if you think about trying to find empirical evidence for hybrid zone movement over long time spans, then you could look for such a genomic footprint of hybrid zone movement. You would predict that to be present on one side of the hybrid zone. So now I'm going to introduce the study system that I work on briefly. These are triturus newts. This picture is a little bit sexist, I realize, because these are all males. But for a reason, they are called the crested newts because the males during the breeding system, they develop this kind of denticulated crest. Now these are marbled newts. They are also in the same genus. They are the main sister group, crested newts, marbled newts. So 7 species of crested newts, 2 species of marbled newts. Marbled newts have like this smooth crest. They have this marbled pattern on the top. And they have a black belly, whereas these crested newts have like this fiery orange belly too. Yeah, kind of like an aposematic signal, like don't eat me, right? So these are the triturus newt species, and this is where they are distributed. So all these different colors represent different species. You have the marbled newts here in green, and the crested newts, all these other colors. So as I said, the marbled and crested newts are the most distinct groups, the deepest level of hierarchy in the genus. They actually overlap in a large region of France. So this is Europe, by the way, and they are also a little bit in adjacent Asia. And then all these other colors are the crested newts. What you see is where marbled newts or crested newts meet each other, you have these very tight turnovers, geographical turnovers, and everywhere where these species meet, they form these neat hybrid zones. So, and these hybrid zones, we already had some ideas that they were in fact moving. So I'm going to present two case studies briefly, looking at these hybrid zones in more detail, and we're going to look for a genomic footprint of hybrid zone movement. So first we're going to move to Western Turkey. Let's zoom in. I switch colors, and I realize that green and red are not the most suitable colors to use. I apologize, historical reasons, and yeah, changing all these colors is a bit too much work. Anyway, here we are. So this is Turkey, Western Turkey. So this is the Bosphorus. Istanbul is here, just to give you an idea. And you see the outline of two actually cryptic species of crested newts defined based on a very small number of nuclear DNA markers. And here, in this part, we already had an idea that something was going on, and that could point towards movement. So what you see here, all these dots, are the sampled populations, and they are colored according to the mitochondrial DNA type that is present. So what you see is that already in this part of the range, you see the green mitochondrial DNA type present in the red species. Also, if you think about comparative phylogeography, there's something interesting going on. So these are actually not crested newts. These are smooth newts, two different species, and they show the exact same asymmetric mtDNA introgression in the exact same region. And also it seems to fit with what we think we know about the geological history of the region. So this here is the Black Sea, this is the Marmara Sea. Currently those are connected through the Bosporus, but there used to be actually an ancient waterway that ran approximately here and connected the Black Sea to the waterway. So Istanbul, the Bosphorus, actually originated relatively recently. So there would have been a moment of opportunity that one waterway closed, the other one opened, where secondary contact could be accomplished and they could move, basically. And that's also exactly where you see syntropy of these two distinct mtDNA types in the same populations. Now, what we did is we started looking for a genomic footprint of hybrid cell movement in that area. We sequenced about 50 nuclear DNA markers, which is actually quite a small number. And we just looked at the genetic composition of these individual populations. So I'm going to explain what all these symbols mean. So you see these yellow stars here. Those are highly admixed populations. So they are basically defining where the hybrid zone is here. So this is around the city of Bursa, if you know it. That is where these two species currently meet and hybridize. Then you see these blue dots. Those are populations that are mostly one species, but still contain clear genetic signature of the other species. And what you already see is, you see that on the east rather than on the west. There's an asymmetry here. And then finally, you see these crosses. Those are simply populations that belong to one species, but contain at least one allele of the other species. And again, you see that there's an asymmetry in space here. So that just shows you present absence, but let's now look at the intensity of introgression of nuclear DNA. Yeah, so what I'm going to plot for you is ancestry versus heterozygosity of individual newts. So ancestry is just the proportion of belonging to one or the other species. If you are 1, you are fully red. If you're 0, you're fully green. And heterozygosity is the fraction of markers that has a green and a red allele. So if you are 0, then all your markers belong to either one or the other species. If you are 1, that means that all your markers are red and green. So a full green species, a fully green individual would end up here, fully red individual would end up here, and a pure F1 hybrid would end up here. So let me now plot the data. That's about 500 newts or something. And what you see is, again, an asymmetry. So many dots are overlaying, by the way, so this is not a single individual, it's many. But what you see is that this overall red newts are kind of being pulled over towards the green side, and much more than the opposite. So there is a geographical asymmetry, again, here in introgression. And then finally we conducted geographical Klein analysis. So what we did is we arranged our localities in transects. We actually studied one transect across the Bosphorus and one across the hybrid zone near Bursa. So the end points of the clients are the same, and this is the kind of like the area of focus on both sides of the Marmara Sea here. So these are the results. What this plot shows you is this thick gray line. That is the overall genomic transition from one to the other species. So what you see here is, this is super steep, it's very rapid, it's very narrow in space. And then you see these thinner clines, those are individual markers. You see the solid red cline, that is the mitochondrial DNA. We already knew that was displaced towards the east, right? And then you see these dotted lines, those are all individual nuclear DNA markers. The ones that are black do not deviate significantly from the actual genomic average, but the ones that are colored do. So those are the ones that are interesting. There are 5 in total that are significantly displaced, and they are all displaced towards the east. So that's unlikely to happen by chance, right? So if you take all this together, we do have evidence here of a moving hybrid zone based on the genomic footprint. A hybrid zone that is moving towards the west. So I'm going to briefly talk about another hybrid zone situation. So this is on the Balkan Peninsula, and you can already see, let's zoom in, there's something odd about the distribution here. You actually have a range segment of one species that is disconnected from the rest of the range of that species. So that is kind of like an enclave. And how could that potentially originate? Well, one thing would be long-distance dispersal. Well, newts are not known for their dispersal capabilities. The distance is about 80 kilometers, the gap in between. So that's just unlikely to happen. But what is more likely is that it basically is an example of incomplete species replacement. So we have basically newts moving around that kind of distribution relict and cutting it off from the main range. And because these newts hybridize, we can test that because we can look for the genomic footprint of hybrid zone movement you would expect. And we already know that mtDNA introgression is rampant in this case. So again, we sampled lots of populations for these 50 nuclear markers in and around the enclave. And here you see their genomic composition based on the overall genetic makeup. And what you can see, we didn't make this distribution pattern up. It is real. There is this core of a blue species that is disconnected from the rest. And you see that there are a red and a green species meeting here. But particularly in this green species, you see little pie slices of blue. You know, blue genes are present well within the range of this green species. This is a bit of a complicated picture. Let me explain. Don't panic. So you see again these dots, those are the sampled populations. The central color of the color of those dots represents the mtDNA type present. And you see Thyssen polygons, so those contain all the area that is closest to one population than to surrounding populations. And then the edge of these polygons represents the species that is genetically dominant there, so of which you have the highest proportion of genetic fragments. And then the inside of these polygons is colored based on how many alleles of the blue species there are. So if you are completely blue, you are completely the blue species. If you're completely white, you're completely one of the other species. And what you hopefully can see that there is some blue shading away from the actual hybrid zone between the species. Here you see again this ancestry and heterozygosity plotted. So it's very two-dimensional in this case. Turkey was easy, just a straight line. Here it's a little bit complicated. So these symbols represent whether populations are actually adjacent to the hybrid zone or very far away from the hybrid zone in the core range of the species. And what you see is that we're going to focus on the green species. We're going to ignore the other ones. You actually see many populations, individuals from populations well away from the hybrid zone that still have blue alleles. Now finally, I also want to do geographical cline analysis, because again, it's very two-dimensional, so that's a bit tricky. I have to simplify it. So what I did is I calculated where the contour line is where they are basically 50-50 one or the other species. So that's kind of like the theoretical hybrid zone, and then determined the distance of each individual population to that line, the minimal distance, so that you can basically collapse this into a one-dimensional situation. So here you see the geographical clines of these different species pairs, and again we're going to focus on the green and blue one. I'm going to lift that out. What you see here is basically this would be the situation if the cline were symmetrical, but it is not. In fact, there is kind of like this tail tapering off into the green species. And again, that is basically, that represents blue alleles being present in the green species. So again, we have several analyses pointing towards this genomic footprint of hybrid zone movement, in this case to the east. So you may notice this blue species is actually being invaded from different sizes, so it's kind of being squeezed out here. So what I'm doing now is basically looking into this situation in much more detail, with also much more genes, only about 6,000 or something. So it has been my intention to publish this for years already, and I should be doing this at a certain stage. Anyway, what are the implications of this of these case studies? Well, just from the point of historical biogeography, I predict that hybrid zone movement over a long time is simply much more common than we have appreciated so far. Also from the perspective of conservation, we are basically creating hybrid zones everywhere. We are driving hybrid zone movement everywhere. We are introducing species into the ranges of other species with which they can hybridize. And finally, if you think about speciation research, this hybrid zone movement is actually separating a lot of the stuff that basically doesn't matter in terms of species integrity, that is free to integrate, from the stuff that actually matters and that actually makes up the species boundary. So I hope that was clear. Thanks for your attention. Thanks to my co-authors. Thanks to the people who paid for all this stuff. And let me know if you have any question.
All right. Who has a question? Yes.
I was wondering about the population that was isolated around. Is there any geographical, some characteristics that make that end up being isolated, like when they...
No, we don't know, but there doesn't appear to be. Yeah. Okay. So these species do differ perhaps in their tolerances, although we haven't really been able to quantify that yet. No. But there's nothing obvious there. It's not like it's a mountain surrounded by lowlands or something. It's not like that.
Okay.
Hi, thanks for your presentation. I have a question, but I have no background about population genetics. I'm wondering which nuclear genes you choose to do research.
So we did not set out to find particular nuclear genes. We just wanted random stuff spread across the genome. So we just took a transcriptome. We looked at 3' UTR regions of genes, so in principle non-coding, and just took it from there.
So do you consider low single copy gene when you...
Yes, we did make sure that they were not paralogs or something.
Yeah, also how can you detect introgression?
Because there is variation between the different species.
Yeah, so how can...
You know the overall genetic makeup of your species. You can take populations, for example, far away from the hybrid zone and consider them unlikely to be affected by gene flow. Then you can determine which alleles are diagnostic for one or the other. And then you can look at the distribution of those alleles.
But you only have 50 genes.
50, yeah, it's not much, but still it was enough to already see a clear asymmetry in the introgression pattern.
Okay, thank you.
Can you also date back the movement of these hybrid zone shifts?
So that's a little bit fuzzy. Yeah. We don't really know when they met up We can kind of show the secondary contact must have been approximately here, because that's where you see the most far inside the range the introgressed alleles. But no, yeah, that's just super fuzzy.
There are actually 2 questions online. So the first person says, really interesting talk. What do you think is driving the asymmetric hybrid zone movement from the first case study you showed?
So for the first case, we really have no idea, because we didn't even know these species existed until not that long ago. So for the case on the Balkan, we have some idea, because we know that overall, what the species that's doing the expanding is relatively more aquatic. So although, you know, climatological reconstructions may not be so high resolution, it seems to be that a relatively dry adapted species is losing out to a relatively wet adapted species.
Okay, and then the second question is, what is it about newts or amphibians that hybrid zones are so easy, between brackets, to see? What about other taxa?
Yeah, I don't know. I think they just take it easy in terms of speciation. It just takes a bit longer. Yeah, it is true that amphibians seem to be doing quite well in having hybrid zones. I don't know about other taxa that much. There are obviously hybrid zones in other taxa, but I look at newts, and that's why I present several newts. Yeah.
Could it have to do with the genome size?
I doubt it. I doubt it. I don't see how that would drive it, but yeah.
What about the lack of dispersal?
Again, they are meeting each other just as well, and no, I don't see how that would drive, say, a propensity to having hybrid zones.
Just thinking, if we think about other taxa and we see less hybrid zones potentially, it could have to do, you know, if you compare it across the tree of life with something related to dispersal, I would think, possibly.
Yeah, maybe, but they're meeting up all over the place. So I get the impression that being isolated, they are not more isolated than other species or for longer time, say, which would be the real reason driving, yeah, genomic incompatibility accumulation, for example. Yeah.
Okay. Any more questions? Or? Good. Then thanks a lot. Very intriguing.
One more question.
One more last-minute question. Thanks for your talk. Do the hybrids have preferences to mate towards one of the parental species or towards both? And does this have an influence on the direction of the introgressed alleles?
So we don't know. There has been very little stuff done. Preferential, definitely not. In terms of viability, what you could think about, there is limited data, but it shows that there is basically no bias in viability of back cross direction, say.
Cool. Thanks also for the questions online. Good. Thanks. We're at the end of the colloquium of 2 days, and I was asked to wrap it up. I didn't have a lot of time to think about this, so I basically wrote down some random thoughts. Maybe do you have the pointer? Which came up watching all these talks. So, I mean, we learned a lot about dispersal. We learned a lot about different systems and how how people in these fields investigate dispersal. One interesting thing is that people online, especially, they would only come in for one talk and then leave again. So it seems generally people are quite system specific, maybe. Whereas we also tried with this colloquium especially to think across systems. So can we learn from other systems to understand our system and the role of dispersal in it a little bit better? So I I mean, feel free to go. It's not going to take that long, I think. But I would like to start a discussion based on my random thoughts and then hear about your random thoughts about these last 2 days. And in that way, maybe bring it together and see what we got out of it, besides very nice talks, of course. So we saw already, many talks started with this. Some species are everywhere. Why? Is it dispersal, or is it the environment acting against species being able to be everywhere? And I guess in many systems, dispersal plays a role, we concluded. Maybe not in all systems. We learn about why dispersing in the first place. Often the motivation might be that you need to get away rather than the motivation that you want to go somewhere. And we've seen that there's a role of both genetics on dispersal ability as well as environment. And environment is changing. So there's both evolutionary components playing a role as well as ecological kind of components, which makes it a very complicated process, but it also links these two fields and how we think about this. The motivation to leave, we saw, is often context dependent, so it depends on the environment. We learned also for fungi, for example, that it might be disturbance, it might be competition happening, therefore I have to go. But of course, dispersal has a cost, so that needs to be, well, maybe the organism somehow weighs these two things before leaving a certain place. But then the environment is so complex and is also so different for different organisms and different groups. And especially if we think of plants and plankton and fungi, maybe like things that don't actively move, they depend on other things for their movements, or they depend on a host or a vector, or they're entirely dependent on ocean currents, or mostly, or things like that. So that adds another dimension to this motivation to leave, right? Maybe it's not motivation, maybe there's just a current passing by, or an animal. We learned that dispersal has been studied a lot from theory and experiments, and we learned a lot from that, but we lack a lot of information from the natural world. It's complicated to measure dispersal, distance, tracking. In some organisms that's simply not possible. We can look at gene flow, but genetic structure is influenced by so many other things as well. So we thought, can we think about dispersal proxies and use a trait-based approach, right? So then the question is, which traits and why? And do we find similarities across groups? Things, proxies, trait proxies that we can use. And then the difficulty that comes with traits is that they may be correlated to dispersal or range size, as we saw in Adriana's talk. But maybe they did not evolve for the purpose of dispersal or getting away. So how do we disentangle these things? Do we need to know the reason they evolved or not? And also, many of the selection pressures may have changed, right? Because that's dynamic as well. So I think these were my random thoughts. Now it's your turn to comment on this and maybe think of, ways forward. Or, oh good, you have already. Good.
I like how, thanks for this is great, but I really like, Renske, how you were talking about the reasons for dispersal, and it reminded me, very random thought, it reminded me how in social science, when they discuss human migration, they talk about push factors and pull factors. And I wonder if that
Good, nice, yeah. Any other thoughts? Or it can also be less philosophical, right? You can also, I'm also curious to hear what you learned from these last 2 days. People online as well, by the way. Maybe there's if they're not all gone already. Anyone? If not, that's okay as well. We will hopefully get back to all these thoughts and somehow make use of them in a paper that will come out of this colloquium or workshop as well. Not on all these random thoughts, but some of them. We have one thing.
Here. This is from Adrian. I learned that dispersal seems highly context dependent, although there are general patterns. So I think this is also what we wanted to accomplish this week, to try to find the general patterns and also try to see how the things also are species dependent, context dependent, and also system dependent, which is going to be the discussion of tomorrow about dispersal in different like marine, terrestrial, below ground systems.
I think what I learned, especially from this meeting, is that you have different ways of looking at dispersal. I mean, and you remember when I started my talk, it's about population level patterns and processes, or a more mechanistic approach. But then, amongst you came up with this kind of more historical approach, biogeographical approach. And I don't have an answer, and I even don't know whether it's a question, but I was just wondering, what can we learn by merging those two fields? So this kind of patterns that have been emerged at large spatial and temporal scales, where you have things going on like speciation. Speciation as the ultimate response towards dispersal limitation or not, and whether we can use those insights to scale down maybe to more local processes these days, or vice versa. Can we actually learn from our experiments that we have been doing at variation within species, local adaptation, to understand speciation events? And especially what you mentioned already here is also this kind of dynamic feedback between both the last sentence there. I mean, because the selection pressures also change because, I mean, dispersal, so actually it's interesting because dispersal is also the process that generates, that continuously changes selection pressures, because the organisms are moving themselves, and it's not the environment which is moving. The organisms move to different environments. So in a way, like the hybrid zones, well, if there would be less non-neutral, for instance, I mean, simply because the movement of some alleles and the environment is changing, then essentially because these hybrid zones or these alleles are changing or moving in the landscape, this might have an impact on the future trajectories of the species. But I don't have it's a random, you ask for random thoughts, and this is a random thought. Yeah.
No, that's perfect. It's inspiring to me, at least. Any other random thoughts? Otherwise, I'll hand over to Adriana to to say a final goodbye.
Thanks. Well, I'm happy that people are still here and people online. I think we learned a lot from everyone, and actually it was very interesting also what you said, that people stay online around 10-30 minutes possibly to stay into the ear system, because I think we should still learn a lot from each other. That I really learned a lot about like fungi dispersal, even for, like plants, everything. It's really interesting, and I think it's something that move forward the field. Well, I guess this is everything like for today. We are going to have another day tomorrow for our workshop to wrap up, and possibly in the future we are going to write a very interesting paper about this, about the work of this week. And I guess everybody, well, I hope everybody is expecting to read it. Yeah. And thanks a lot. See you next time.